Right. Aki is not here. Bob is not here. Bonner is not here. Clay is not here. Dawson is not here. Ellis is not here. Castro is not here. Green is not here. Harrison is here. Henson is here. And WSU here.
Yep.
WSU here. James is not here. Kesterson is here. King is not here. Lewis is not here. Linda Cruz is not here. Morrison is not here. Grace is not here. Ward is not here. Wilcox is not here. Wilder is not here. Parks is not here. Ellis is now here. Let's see. Let's see who's online. Most of you have. So don't forget if you're online to send the attendance words. Just as some reminders. Again, Friday I'm not holding office hours because of things I have to do. Today, you can come into lab, or you can come join us virtually, or you can do it on your own. Like I said on Monday, I recommend before you make that decision, take a look at the lab and see if you can do it on your own, or if you think it's going to be a hassle. If you don't feel like doing it on your own, you can come in and we'll do it as a big group. So yes, take a look at that and then come in if you want to. Wilcox is here. What are the other reminders? Oh yeah, the other reminders, next Monday, remember we're meeting in person only, so not online. I mean, good grief, I got that backwards. We're meeting online only, not in person. Like I've been saying, if for some reason you're really opposed to that, let me know. I'll just come in anyway. What else? The exam is due on Sunday. And today we're going to finish chapter 9, which is the last part of this exam. The study guides are the quizzes are the study guides for the exam. As always, with any assignment, you can meet me online and we can go over the quizzes. Then I can tell you why your wrong answers are wrong if they are wrong. So not only will you get your 5% boost to that assignment or any assignment you need it for, but also then you'll have a better way of studying because you'll know all the right answers for the study guide, aka the quizzes. So let me know if you want to do that. And Dawson is here. And WBSU here. So I guess that's it for the announcements and reminders. Oh yeah, and again, independent work. Please do independent work. It's a full 100 points. The whole course is worth 1,000 points. So it's worth an entire letter grade. Meaning, if you somehow only got 500 points this semester, which is failing, but you did all your independent work, that means you'd have 600 points, which means you would pass. It's enough to make you pass. But then again, conversely, if you had 600 points at the end of the semester, if you would have had 600 points at the end of the semester but didn't do any independent work, then you're going to fail. So get your independent work done. Does anyone have any questions before we get started? All right. So again, today we're going to jump into, we're going to finish chapter 9, and then we're going to jump into the next chapter that we're going to cover, which is chapter 11. All right. So the first word for attendance is fighting, like the verb fighting, or the adjective if you were to talk about somebody, which I am. So fighting is the first word for attendance. All right, and here we go. Again, we're almost done here. Whoa. Pretty good if I showed it. Why is it not showing up? Excuse me. It's showing up on this end, but not on the other. There we go. Anyway, so yes, we're almost done. We're still talking about viruses. This is the last virus we're going to talk about. There's not going to be any questions about it on the exam, so I'm going to go through it a little bit quickly. And then after that, we're finally going to talk about prions, which is not a virus, but it is an infectious agent, and it's not cellular, so that's why we're going to talk about it. Anyway, Let's talk about AIDS really quickly, AIDS and HIV. AIDS stands for Acquired Immunodeficiency Syndrome. I'm not going to ask you that. That's the name of the disease. The disease is caused by a virus called HIV. HIV stands for Human Immunodeficiency Virus. I'm not going to ask you that. But what you might need to know is what type of virus this is, because it's an interesting type of virus. It's called a retrovirus. This particular retroviruses are RNA viruses. I might ask you that. So again, you might need to know, you might need to know that AIDS is a retrovirus. But more importantly, if I do ask about retroviruses, you would need to know that they are RNA viruses, right? They're not DNA viruses, they are RNA viruses. But they're interesting because they reproduce with a DNA molecule. So even though they're an RNA virus, their reproduction is through a DNA molecule. And that's unlike anything I've told you before. And again, I'm putting a check mark there because if I was going to ask you any questions about retroviruses, these are the things you're going to need to know. And the reason this is interesting is because it's the reverse of the usual DNA to RNA to proteins, right? Or in this case, since we're talking about viruses, going from DNA to RNA. In this case, we're going from RNA to DNA. And this is accomplished by something called reverse transcriptase. That's the name of the enzyme. So again, as far as the exam is concerned, if I ask any questions, you should know this about retroviruses. Again, they're RNA viruses. They reproduce with a DNA molecule. Therefore, logically, they must go from RNA to DNA, right? Because if they are an RNA virus, but they reproduce with a DNA molecule, then it has to be going from RNA to DNA. And of course, you're going to need a special enzyme to do that. And again, it's in the name, so hopefully that helps you remember. Reverse transcriptase. And it's exactly what it sounds like it does. Because remember, what does transcription do? It goes from DNA to RNA. So what would be the reverse of that? It would be RNA to DNA. So are there any questions about this slide before I give you some visualizations of what we're talking about? All right, this picture is not from your textbook. This is from the old textbook. This is what a typical HIV virus looks like. You can see the membrane on the outside with these protein spikes as usual, right? That's kind of common for an animal virus. You can see the protein capsid enclosing the genetic material. And in this case, because it's HIV, the genetic material is an RNA strand, technically 2 of them, and then technically 2 reverse transcriptase enzymes. But I'm not going to get that specific, right? I'm not going to show you this picture and ask you to name the parts. You don't need to memorize the fact that HIV is enclosed in a membrane and also a protein capsid. You don't need to memorize the fact that there's 2 RNA strands there and 2 reverse transcriptase enzymes. But there's still more to show you. Are there any questions about this so far, though? Okay, here we go. This is kind of how it works. So, of course, I'm missing a portion of the picture, but imagine, like you've seen before, the genetic material is inserted from the virus into the cell. And this is what we're looking at is the cell here. Here's that viral RNA. Here's that reverse transcriptase that reads the RNA and does reverse of what we're used to. It reads that RNA and looks at those complementary nucleotides and brings in the complementary nucleotides and makes a DNA strand out of it. And then it goes through some more magic, so to speak, and turns that single DNA strand into a double DNA strand. And this is where it gets nasty again, too. Then that double DNA strand is then incorporated into your genomes. That part should look familiar, right? So when we talk about things like herpes, as one example, that is something that happened, right? The DNA from the virus actually got incorporated into your own chromosomes. That way, every time you went through mitosis, you are not only reproducing your chromosomes, but also that virus. But anyway, that's where it lives, right? And like I said in the previous slide, this is an interesting virus because it reproduces through DNA. And not just any old DNA, right? This is your DNA, in a sense. Yes, it was viral DNA, but then it is incorporated into your chromosomes. So every time this virus reproduces itself, it's because your RNA polymerase just attached to your DNA, just like it normally does, and it reads it, it makes the mRNA. mRNA comes, does the normal thing, goes to this splicing and Cacotail, comes out of the nucleus, and then just as you've already learned about translation, that happens again, right? That RNA is translated. That makes the new viral particles that get put back together. And then finally, the new virus, then leaves the cell and then can go and infect other cells. And of course, this picture on the right is what it looks like from a scanning electron microscope. And again, I'm probably not going to ask you any questions about HIV on the exam, and if I do, I'm certainly not going to go into any of these details. The big thing here is that you understand how reverse transcriptase works, or how reverse transcriptase, how that works. Because it's the opposite of everything I've taught you, which makes it slightly interesting. Anyway, any questions about that side? All right, if you download the PowerPoint, you can watch this little animation right here that kind of shows you how it works. Basically everything I just showed you, but animated. And I'm definitely not going to ask you any of this, but I'm going to talk about it really quickly anyway. It's already on the PowerPoint, so why not? How HIV works, right? HIV kills white blood cells, and white blood cells are important in your immune system. So basically, it kills your body's ability to protect itself, which makes it so bad, right? When the old textbook was written, technically there was no cure, but it was slowed by 2 categories of drugs. Things have changed a little bit now. You can look that up for independent work. Again, for one point, you can just answer the question, is there a cure for HIV? I looked it up, and here's the answer, right? Copy, paste it, give me your source. And if you want to, you can write a book or a paper about it. In your own words, you write about it, summarize it in your own words. Don't use AI, don't reword it, just write about it in your own words. Anyway, really quickly, really quickly, I'm almost definitely not going to ask you about this on the exam. But both of these 2 categories of drugs that are against HIV, they both interfere with the reproduction of the virus. So it's not necessarily that they're killing the virus, they're just stopping it from reproducing, which eventually means it will die out. But the first one inhibits the protease enzymes, which helps produce the HIV proteins. So I'm going to back up and show you this very quickly. This picture didn't get into it much, but you can see here that this RNA, again, came out, this viral RNA, came out and it was translated into all these different proteins that make up the virus. So there's these enzymes that are not shown in this picture that are doing this assembly, right? It's not like these things are just made and they come together magically, right? There's enzymes that do this. So again, the first category of drugs basically stops it from doing that. And you learned about enzymes, you learned about enzyme inhibitors, and that's exactly what it does. It inhibits those enzymes, therefore it stops it from reproducing. The second category of drugs inhibits the actual reverse transcriptase. So this is really kind of stopping it more from the beginning. This is a very big thing about form and function. And I'll just show you the picture, this will make more sense. This is how it works in a nutshell. This is what the T looks like, as you should be already familiar with, right? This is a T nucleotide. You've got your sugar, phosphate, that's not shown right there, and a base. But what they do, when you get this drug, you have this thing called AZT, which is very similar to T, right? You can see the chemical is very similar. Can anybody see the difference? And I'm looking to try to remember. Oh yeah, there it is. So here we have an OH group, and here we have an N3 group. And that one little thing, remember, this is a good reminder for something you're going to need to know for the exam. Remember, nucleotides are covalently bonded to each other from the sugar to the phosphate, sugar to the phosphate. You can't see that right now. So the sugar right here, this OH, gets bonded to the phosphate. But since this has an N3 instead of an OH, it cannot do the bonding, right? Therefore, it cannot make a strand of nucleotides. And that's how that works, right? So, and again, it's this great form and function thing. Your cellular machinery doesn't know any better. When it's like reading the code and bringing in nucleotides to make in that new DNA strand, it says, oh, we need a T. So it's bringing in whatever T's are floating around in your cytoplasm, and it thinks this is a T. So it brings it in, then that's the thing right there. Just because of that one different group, then you cannot have anything covalently bonded to it. Therefore, the whole strand is, it stops production. And again, this isn't important for the exam, but thinking about how this works with the sugar to phosphate bonds, that is important for the exam because you do need to understand how nucleotides are bonded to each other. Anyway, any questions about that? Okay. Last thing we're going to talk about for this chapter, before we jump into the next chapter, chapter 11, we're going to start with that today, is something called prions. And again, I'll tell you there's probably not going to be any questions about this on the exam, and if there are, they're going to be very few. Well, we've actually talked about prions before, so this is actually our second time talking about them. These are infectious proteins. right? So they're not viruses, and we know viruses aren't alive. Viruses are just genetic material encapsulated in protein. We've talked about bacteria before, which also cause infections because they are alive. Those are cells that infect you. But prions, just like viruses, are not alive. They are simply proteins, but they're infectious proteins, and they cause brain diseases. Specifically, they are a misfolded form of normal brain proteins. And what happens in a nutshell is these misfolded proteins come in contact with regular proteins, and it causes them to misfold. And then now you have more misfolded proteins, and they go out, and every time they touch a protein, they cause them to misfold. And this causes basically holes in your brain. So some examples of diseases caused by prions are scrappy in sheep, chronic wasting disease in deer and elk, which some people around here might know about. If you're into hunting, you've probably heard of that. Actually, I've even heard about it on the news recently. Mad cow disease, you may remember that if you're old enough. Creutzfeldt-Jakob disease in humans, and then Kuru, and that's the one I mentioned previously when we talked about this, because that's the one that was caused by cannibalism, or is caused by cannibalism when people eat other people's brains. So again, for independent work, you can look any of these up and write a paper. How does it, what happens to sheep? They get scrappy. What happens to deer and elk? They get chronic wasting disease, so on and so forth. How prevalent are any of these things, right? Anyway, again, I'm not going to ask much about it. So are there any questions about that? All right, so for this chapter, as far as we're concerned, that's the end of it. Again, this PowerPoint was based off the old textbook, so I included this, which talks about the evolution connection of emerging viruses. I recommend reading it. It's really good information, but I'm not going to ask you any questions about it. So now I'm going to pull up the next PowerPoint for chapter 11, which is a good time to remind you that we just finished chapter 9, skipping chapter 10, and going to chapter 11. But I highly recommend reading chapter 10. None of the information in chapter 10 is going to be on the exam. But to understand the information in chapter 10, you're using information that you've learned and that you're going to be tested on exam 3. So that's how it would be useful. And if I get a chance, it doesn't look like I will, but if I get a chance, I will create some sort of quiz for chapter 10 that you guys can take for extra credit. Anyway, here we go. Chapter 11. The next word for attendance is Quaker. Like the group of religious people, or Quaker Oats, or Quaker Steak, and what's that place called? Quaker Steak and Lube up in Southridge. That is the next word for attendance, Quaker. Okay, here we go. Another thing I want to point out is, again, I think your old textbook did a better job of giving you this information. So I'm sticking with the old textbook as far as the order in which I'm giving you the information. So another thing to point out is we are combining chapters 11 and chapters 12 into this one PowerPoint. Because again, I think your old textbook did a better job. But your old textbook wasn't free, so you get what you paid for. So yeah, so this is all about evolution. Let's talk about why evolution matters. First of all, can anybody even tell what this thing is on that burger? Tomato, correct. That is a tomato. And what your textbook, the old textbook was getting at with that picture is that's what tomatoes used to look like. That's just what wild tomatoes look like. And then humans used artificial selection, which is a type of evolution, to make them into what we see today. Now, granted, there are still tomatoes like that, like cherry tomatoes and things like that. But I think usually when people think of tomatoes, they think of the larger slices. And that's why. It's because of evolution. That is an example of evolution. Granted, it's human-caused evolution, but that is evolution. This one's a little bit harder to guess, but does anybody want to venture a guess of what's happening with that kid?
Head lice.
Good, that is a great guess, and you are correct. Yeah, kid's got head lice. And later in this chapter, we're going to talk a little bit about how insecticide resistance works. And basically, treating head lice it's an insecticide that you're putting in your scalp. But because of natural selection, there is something called insecticide resistance. Same thing with head lice too, right? So if you, long story short, and we'll get into more detail later, when you apply the treatment to kill all the lice on the head, if it doesn't kill all of them, then the ones that are left are the really strong ones, right? Because there was something genetically about them that made them unsusceptible to that pesticide. And therefore, when they go to reproduce, and that new population that comes out is now resistant to the pesticide. So it could be a bad thing. We'll talk about it later. This one's a lot less, what's the word I'm looking for? Harder to guess. Obviously, that's two cheetahs. I'll just tell you what your old textbook said about that. The cheetah may be racing towards extinction. Many endangered species are doomed by the lack of genetic diversity. So, and I'm going to go into more detail later in this chapter, but cheetahs almost went extinct twice. Their numbers are better than they used to be. However, because they almost went extinct twice, they have very little genetic diversity. And later in this chapter, you're going to learn about why that is bad. Like, we need genetic diversity for our species survival. Or in this case, they need genetic diversity for their species survival, but we do too. Anyway, any questions about that side? All right, here's the overview. And again, this overview is the way I'm teaching it. It's going to be slightly different than your textbook. So look, for example, the fact that we're starting off with the diversity of life, which is actually chapter 12 in your textbook. Then we're going to come back and talk about some stuff that's in chapter 11. Specifically, you're going to learn about Charles Darwin and the origins of species. That's his book. We're going to talk about the evidence of evolution. We're talking about how natural selection is one of the mechanisms for evolution, a big one. That's one we talk about most and study most. We're going to talk about the evolutions of populations, which is what this week's lab is basically about, microevolution. And then we're going to talk about the mechanisms of evolution. So here we go. First, let's talk about the diversity of life, which again is chapter 12 in your textbook. Here's a word you might need to know: taxonomy. That is the branch of biology that deals with identifying, naming, and classifying species, right? So when you hear somebody say, hey, we found a new species of this or a new species of that, that's taxonomy, right? They deal with all that stuff. We have somebody in this building, his name's Dr. Runke. He deals a lot with taxonomy, the taxonomy of tapeworms, actually. He's found a lot of species, new species of tapeworms. But I'll talk about that later too. Anyway, that's what taxonomy is. Are there any questions about that? All right. The next thing we'll talk about is the Linnaean system. You need to know about the Linnaean system. This is the method of naming species. So it's not random, right? We can't just say, I'm going to call this thing Bob, right? We have a system for it, and that is called the Linnaean system. It's a hierarchical classification of species into broader groups of organisms. And we'll go into more details later, but that's just, like, for example, when you say a human is a Homo sapien, it's not just the end of it. We also get more specific, we get broader and broader, right? We're also mammals, right? We're also animals. We're also eukaryotes, right? So that's what we mean by that. And humans just do this naturally, right? We are naturally into classifying things into broader and broader categories. Like, I drive a Toyota Prius Prime, and you get a little bit broader, which is also a Toyota Prius. A little bit broader, which is a Toyota. A little bit broader, which is a vehicle made in Japan. A little bit broader, which is a vehicle, right? So you can get broader and broader and broader. And again, we'll jump into more details with that later. First, let's talk about naming and classifying the diversity of life. So again, we're going to get into this idea of the Linnaean system. A species is given a two-part Latinized name, aka a binomial, so you need to know this. The first part of the species name is the genus. The second part is the specific species. You need to know that. So a genus is going usually, not always, sometimes one genus only has one species. But a genus is a group of closely related species, right? So there's a bunch of species, not a bunch. There's sometimes several species under one genus. In your book, the old textbook gives the example of the genus of large cats called Panthera. Throughout this discussion, I'll give you some other examples, some species examples that fall under Panthera. You definitely don't need to know that example, though. So let me write it next to that. I'm not going to ask you what Panthera is, or I'm not even going to ask you to remember that it's a genus. But anyway, yes, the first part is the genus. The second part is the species within the genus. So you need to know that. So again, we are Homo sapiens, meaning Homo is the genus. We specifically are sapiens. And again, for independent work, you could look up some other examples. What are some other genuses what are the other species within the genus Homo? Of course, none of them are alive anymore. We're the only ones alive, but there were some other ones. So the two-part together, the genus and the species, that is the scientific name, right? And again, just as an example, the scientific name for a leopard is Panthera pardus. So that is the genus, that is the species. You do need to know that, not Panthera pardus, but the first part is the genus and the second part is the species. I will give you a random species name that I would assume you've never heard of, or maybe that I made up. And then you have to tell me which part is the genus, which part is the species. Another thing to note is when you write these scientific names down, they're always supposed to be in italics. And the first part, the genus, is always capitalized, and the second part is not capitalized. Those are all things you need to know for the exam. So the question is why? Why do we even have this, right? Why not just call humans, humans, and deer, deer, and dogs, dogs? Like, why do we do any of this? Any guesses? All right. I wish I didn't have all those letters up there, but pretend like you can't see that. What's this thing up there? Does anybody know?
Mahi.
Yeah, it's mahi mahi, yeah. But that's what you call it. That's what a lot of people call it. Back in my day, when I was a kid living in the Keys, the actual word, they used the word dolphin. Like, so if you went to the restaurants in the Keys in the 80s and you wanted to eat that, it said dolphin on the menu. But I think eventually that got to scare people because they thought a flipper, like an actual dolphin. So they stopped using that word and went for the Hawaiian word, which is mahi mahi. But then if you were speaking Spanish, if you're in a Spanish-speaking country, it's called dorado. If you're in Japan, it's called shira, right? So the point here is, this is why we have scientific names, right? Because no matter where you go, you'll have different names for different animals. Or not just animals, but plants, right? Any living organism. So this is why we have scientific names. It relieves the ambiguity. So a scientist from Ireland can talk to a scientist from South Africa, and they'll know what they mean when they use that scientific name. Which is another great independent work topic. Like, for one point, choose any organism you want. Just email me, like, this thing we call it a squirrel, but its actual name is, according to this source, this is the name, right? Any questions about this? Just an example. I'm obviously going to not ask you the scientific name of mahi mahi or the other names of mahi mahi. That's just an example. Here's where it does get a little bit harder, though. Like I said, this whole classification thing is about having broader and broader and broader categories. Right? Everything is categorized. It's not just a species or just a genus. We're categorizing it in larger and larger categories. That's where taxonomy comes in. So, in this example, I'm going to teach it to you going specific to broader, and you need to know it both ways. You need to know going from specific to broad, and then from broad to specific. Geographically, if I was going to give you an example of that, I would say, all right, very specifically, this is our address. Whatever it is, 12, actually, I should know our address. Anybody know the address here? Something, 1212 Fairlawn Avenue. Institute, West Virginia, and I even forgot the zip code. I feel horrible. But that's very specific, right? And then we can get a little bit more broad and just say, oh yeah, we're in Fairlawn Avenue in Institute, West Virginia, 25512. And then you could get even broader, like, yes, we're in Institute, West Virginia, zip code 25512. And you could get even broader and just say we're in 25512 in West Virginia. And you could get even broader and say we're in West Virginia. And then you could say we're in the United States of America. And then you could say we're in North America, right? Same thing, except now instead of talking about geography, we're talking about organisms. So remember, the most specific is species, right? Species is the most specific, right? And then as you already know, you can have a bunch of similar species in the same genus, right? And then you can have a bunch of similar genus in the same family. And then a bunch of similar families in the same order, a bunch of orders in the same class, a bunch of classes in the same phylum, a bunch of phylum in the same kingdom, and the same kingdoms in the same domains. So again, you're going to need to know that, specific to broad and broad to specific. And probably you learned it like this. Most likely, from everybody I know, usually they've learned broader to more specific, and they had to memorize kingdom, phylum, class, order, family, genus, species. So you do need to know that, but also don't forget the domain is actually above kingdom. The domain, kingdom, phylum, class, order, family, genus, species. You need to know that for the example. More than likely, I'll give you the scientific name of a of an organism and then ask you, and then give you a little bit of information in addition to it, and then ask you what class is it in, what order is it in, and you'll know because I give it to you in order. All right, let me use this as an example. For example, I might say, let's see, this is Panthera pardalis. Okay, so I might say the species Panthera pardalis, and then it'll just list all these words in order. Felidae, Carnivora, Mammalia, Chordata, and Animalia, or Eukarya. And then I'll say, which one of those is the family? So then you'll be like, all right, so you see, species is the Panthera pardalis. Then you'll just need to go, all right, so that's the species, that's the genus, therefore that must be the family, therefore that must be the order, right? So you just need to know it in order. Any questions about that? Then if you want, I can give you any of this. If any of you are having problems with any of this, let me know, and I'll come up with some practice questions that maybe you can do for extra credit. All right, but really quickly, a big X through this, because yes, you do need to know domain, kingdom, phylum, class, order, family, genus, species, but you definitely don't need to know specifically about that panther. That's not important. Here's a different picture from a different textbook. Kind of talks about the same thing, just has a different visualization. So once again, you're looking at the domain eukarya, which is all eukaryotes, right, including some protists over there, right? Then we kind of focus in and look at the kingdom, the animal kingdom, right? So now we're just looking at animals. We're not looking at protists anymore, or plants, right? Then we get a little bit more specific and talk about chordata. So now we're only talking about things that have, well, basically vertebrates, but there's a slight difference. We won't even get into that. Then we get into subphylums, which you don't need to worry about. Then we get into the class. In this case, now we're only looking at mammals, right? So we're no longer looking at birds, for example. Then we get specific, even more specific, and talk about the order Rodentia. So now we're only talking about rodents, right? We're not talking about monkeys or primates anymore. Then we get even more specific, and you can see it goes down to the family, genus, species, so on and so forth. But that's a great visualization too, because again, this is talking about a very specific type of squirrel that you can see as it broadens, it includes more different types of organisms. So any questions about that side? And again, a big X through here, because you don't need to know those specific examples of that squirrel. You just need to know domain, kingdom, phylum, class, order, family, genus, species. And also a big old X through that. Yes, there are subphylums. There are also superclasses and suborders. But all that stuff you don't have to worry about because it's a 100 level non-biology major course. But you can look that stuff up if you want. And again, another good independent work topic. Choose any organism and write a quick paper about it. Give me its complete classification, right? So, dogs, dolphins, a specific type of mushroom, whatever it is that you're interested in. Right, just give me the whole classification. Anyway, if there's no questions, let's move forward. Oh yeah, I like this. This is a good thing to point out too. The criteria used to classify species is generally arbitrary. I wouldn't say ultimately, that's what the old textbook says. It used to be a lot more arbitrary than it is, but after you learn about the processes by which the diversity of life evolved, which is coming next, we will then introduce a classification system based on the understanding of evolutionary relationships. So it's not completely arbitrary. When we say something's related, that means things that are in the same genus, for example, all these things, of course I'm not showing these two squirrels, those different species of squirrels have a common ancestor. And then these have a common ancestor, right? So it's all about evolutionary relationships is how these are classified. It didn't used to be that way. It used to be, like the name implies, like vertebrates, right? It used to be like someone said, all right, well, if they got vertebrates, they're related. Or excuse me, vertebrates, they're related. And which it's true they are. But now when we learn more about evolution, we can kind of classify them a little bit better. If you download the PowerPoint, you can click this link there, and I'll show you a little video about taxonomy. Now, here's something very important. You definitely need to know this. I'll take my time on this next slide. You need to know what a species is. A species is a group of actually or potentially interbreeding individuals. So it's individuals that are breeding together. That's a species. And then I like the potentially thing. So for example, I wish I could think of one off the top of my head, but I haven't had any coffee today. But if you think about, even though this isn't a natural thing, they're in zoos. But you have a panda in San Diego and you have a panda in China. They're not breeding because they're separated. They're not going to ever meet up. But they could potentially breed, right? If you were to put them together, they would be able to produce offspring. Maybe that's a better way of putting it. Species is a group of individuals who could produce offspring. One species is distinguished from another by the possibility of matings between individuals from each species to produce fertile offspring. So that's kind of the same thing I said, but let's focus on that word for a second. So again, it has to be fertile offspring. A lion and a tiger can produce an offspring, but the offspring won't be fertile. Therefore, lions and tigers are different species. A horse and a donkey can produce an offspring, but the offspring isn't fertile. Therefore, the horse and the horse and the donkey are not the same species, right? Different species. There are exceptions to that rule. We won't even get into it. You can look into it if you want. So, does anybody have any questions about what a species is? It's very important because we will be discussing species and populations and things like that when we get into evolution. So, it's important you understand what a species is. All right. So, the next word for attendance is Smedley. It's a weird name, so I'll write it out for you. Smedley. Anyway, speciation is another term you should know, and that is the formation of two species from one original species. Actually, I take that back. Even knowing the word is not as important as knowing the concept. We'll talk about the concepts is more important. The point being here is that one species, through evolution, can change into two species. There's different types. The first one we're going to talk about, and you need to know the difference between them, is allopatric speciation. And this involves the geographic separation of a population. Meaning, again, we have that one original species, and it's going to be changing into two new species. And in this specific instance, it's happening because of a geographic separation of them. Technically, this is also broken down into dispersal and vicariance, but we won't even get into that. It's in the textbook, so I included it. There it is. You can read about it if you want, but I'm not going to get into it. Not too much. Anyway, the next type is called sympatric speciation, and this is speciation within a parent species, like we were talking about already, while remaining in one location. So in other words, again, we're talking about speciation, which is when one species turns into two species. And there's two different categories, one of which is called allopatric speciation, which is when they've been separated from each other geographically. And then we have sympatric speciation, which is where they have not been separated. It's that simple, at least for now. We're going to make it a little bit more complicated, but that's a good starting point. So the way I always remember this is sympatric, right? Similar, they're in a similar location. Sympatric speciation, similar location. Allopatric, separate locations. Any questions about that before we dive deeper into it?
Are we talking something like alligators and crocodiles?
Yeah. Because they had originally one ancestral species that did split into those, and then the crocodiles split into different types of crocodiles. So yeah, that's a good example. And also think about all the different, like I said earlier, Homo sapiens are the only hominids still alive, but there were other ones, right? So that we had an original species that split off into all these different types of humans. Of course, all the other ones are dead, like Neanderthals, things like that. So here's a good visualization, I think. This is not from your textbook, but we're talking about allopatric speciation. Think about any animal here. It's kind of hard to see. Man, it's better on the screen. But what we're looking at here, if you can't tell, is we have the Pacific Ocean over here, like Seattle. That's Olympia. Yeah, so Seattle area, the Pacific Northwest. And then on the other side is just regular Washington, Idaho, that area. And what happened is, I don't know if you guys know anything about it, but there's a lot of mountains there, right? The reason there's mountains there is because there's a plate in the Pacific This is a little bit easier. Yeah, that's easier to see. See, there's Seattle area, right? Vancouver, Washington, Northern California, Idaho, all that. So what's happened is this plate is crashing into the United States. And as it does that, it's causing these mountains to come up. So imagine before that happened, that used to be probably just kind of flat land. So imagine if you were a species like, I don't know, some type of deer or elk. I'm just throwing that out there as an example. And you're just roaming this whole area. You're just one species. You're interacting together. You're interbreeding together. You're dealing with the same conditions. Oh, actually, no, let me give you a better example. Let's talk about some kind of tree. There's a tree. Let's say there's some kind of tree species that lives in this whole area, right? And they're living the same situation, right? The same weather conditions, the same climate conditions, the same diseases. It's all the same. Then all of a sudden, this plate comes crashing into North America, and you have this mountain range come up between you. So now you have trees on the left-hand side who are getting a lot of rain, because also now when the weather comes in, it can't make it over the mountains. So it just rains constantly on Seattle. And I'm sure you guys knew it rained constantly on Seattle. Now you know why. It's because that weather can't get over the mountains. Therefore, on the other side of the mountains is basically desert. So now think about that same species of tree. Now you've got the trees on the left-hand side getting a ton of rain, and the other ones on the right-hand side are now basically living in desert conditions. So over a few generations, right, now you're going to have some new species. Because only the ones that can deal with a lot of water on this side are going to survive, and the only ones that are going to survive on the other side are the ones that can deal with a lot of drought. And that is a great example of allopatric speciation. Right? You had one species that was dealing with the same thing, but then they got geographically separated. Now they're dealing with different things. And again, only the strong survive, right? So on the left-hand side, when we say only the strong survive, what we mean is only the ones that can deal with a lot of water. And on the right-hand side, it's only the strong survive means the ones who can only deal with drought conditions. So any questions about that? And again, that's just an example. I'm not going to ask you about the Cascades or Washington or this hypothetical tree example I just made up. Okay, so we'll talk about this. Adaptive radiation. This is when the population of one species disperses throughout an area. So we're still talking about allopatric speciation, except in this case, like so the last case I gave you, that hypothetical example, I was saying the land changed, right? And that separated these two groups of individuals. Now, it's not the land changing, it's just that the population is dispersing and changing, just like what happened with animals. I forgot who asked me, but someone was asking me about, like the white skin from Caucasians. How did that come up? That's because humans migrated out of Africa, and of course we're still the same species, but we changed, right? So somebody had a mutation that caused them to be really light, and it was up in Europe, and there was actually an evolutionary advantage for that, so that whiteness persisted in that population, right? So this is a great example of adaptive radiation. Of course, in the example I just gave with humans, we're still the same species, but again, as we migrated out of Africa, we changed. Not because the geography forced us to, but we went to different geographies. So it's still allopatric because we are still separated, but it's because we had the adaptive radiation. So again, what happens is each of these new individuals, as they're migrating away, they find a distinct niche, which we'll talk about later, or isolated habitat, and they're just well fit to it. So again, just using humans as an example, think about migrating out of Africa. Sub-Saharan Africa is really hot and humid and a lot of trees, and then you go through the desert, right? And you go up north where it's snow covered. So all these different habitats, and then therefore humans changed. And then of course that picture shows different birds. You can look into that if you want. That's even hard for me to read on the screen here. But if you download the PowerPoint, you can see what it is it's one species of birds that have migrated to different places and have different sources of food. And therefore, because they had different sources of food, then their beaks changed to take advantage of the different sources of food. Again, it started off as one species, and then they migrated away, and each of them had different little types of living conditions, and because of that, that changed them every time. Here's a fake example, but it's based on a slight bit of reality. Think about the moths you had. Imagine if those moths were not even in that area at first, right? So it wasn't the pollution that necessarily changed them necessarily. because, the moths were already there. But imagine if the moths came into the polluted area, right? Maybe there was a species of moths and they migrated, and some of them went to the polluted area, some of them went to the non-polluted area. The ones that went to the polluted area are going to turn dark. The ones that went to the non-polluted areas are going to turn, stay light because they've migrated. They went to different locations that have different requirements, and therefore they changed. Is there any questions about adaptive radiation? All right. Here's another interesting way this could happen: is chromosomal errors during meiosis or the formation of a hybrid individual that has too many chromosomes, right? So usually, this is why I said earlier, there's exceptions to the rules. Usually when you mix two different species, the hybrid is not fertile, therefore, it just doesn't do anything because it's not fertile. It won't create other organisms. Sometimes it does, and sometimes that is another way that a new species arises from the mixture of two species. And again, that's not usually what happens, but it can happen. Technically, the word for this, as we mentioned earlier, is called polyploidy. We talked about that when we talked about mitosis and meiosis. And a polyploidy is when a cell or an organism has an extra set or extra sets of chromosomes. So that word, it's not new to you. Any questions about that? All right, here's the picture. I'm trying to remember. This is from your actual textbook, so I'm trying to remember what it is. You have a thin-lipped cichlid on the left and a thick-lipped cichlid on the right. I took notes on here. Let me read my notes, sorry. Cichlid fish from Lake Apuyolque in Nicaragua show evidence of sympatric speciation. So again, they're in the same lake, sympatric, right, same area. This is a crater lake. It's 1,800 years old, so relatively new, right? 1,800 in terms of geological time is very, very new. But there is genetic evidence that indicates that the lake was populated only 100 years ago by a single population of these cichlid fish. Nevertheless, two populations with distinct morphologies and diets now exist in the lake, and scientists believe these species may have been in an early stage of speciation. So there you go. So they haven't technically speciated yet, but they're working on it, right? So there's different types. These are the same species, but they haven't speciated yet. But again, this has only been 100 years. So think about it. You have this new lake 100 years ago, which is really, really, really not long ago in these kind of terms. Brought these fish in, and already, because you can imagine how a lake is, there's shallow parts, deep parts, hot parts, cold parts, right? So they've gone throughout the lake. And they've adapted to their different parts of the lake, and they're already starting to show some differences. Obviously, not just the way they look, but also according to the textbook and their genetics as well. All because they're just in different parts of the lake, dealing with different environments, and natural selection is taking its place, and we're eventually going to see new species because of it. So again, these are different types of fish, but they're still the same species. They can still interbreed and produce offspring. But what we're saying here is eventually they might turn into two separate species in which they could not interbreed and produce fertile offspring. Any questions about that? Again, big old X, it's just an example. I'm not going to ask you any questions about those fish. Okay. That's a good place to stop. We'll stop 4 minutes early. And the last word for attendance is butler. Like the last name Butler. Or the profession. That's it. I'll see you today at 10 if you want to be here, or if you want to be online. Otherwise, I'll see you Friday. See if it actually recorded.